Hi guys, today we will be discussing water distribution and its regulation. Before we get into it, recall that homeostasis is a process by which living organisms maintain a stable internal environment despite external changes. Water homeostasis is the regulation of water levels within the body to ensure a proper balance and prevent dehydration or overhydration. How does body maintain homeostasis? Via a process known as feedback loops. Recall that there are two types of feedback loops, negative feedback loop and a positive feedback loop. Positive feedback loop usually amplifies the initial change, whereas negative feedback loop operates to counteract the deviation from a set point, thereby maintaining the system. It has three interdependent components, a sensor, an integrating center, and an effector. Let's take an example. When body temperature rises above a certain point, the thermoreceptor signals the brain to initiate sweating and vasodilation, thereby bringing the temperature back to the original set point. So here, the loop is counteracting the change, which is the exceeding of temperature above a set point. Suppose we have same individual, now he is suffering from dehydration. His plasma osmolarity has increased. What will happen is that the osmoreceptors will detect this change in osmolarity and will send the signals to the control center of the brain, which is the hypothalamus. The hypothalamus then will carry out the response by releasing antidiuretic hormone from supraoptic nuclei and posterior pituitary, where it has been stored. Then antidiuretic hormone will act on the effector organ, which is the kidney. It will conserve the free water by inserting the aquaporin channels. So how does this ligand exert its effect? To get into it, you need to understand the concept of signal transduction. What is signal transduction? When extracellular signals are converted into the intracellular signals. Here, a signaling molecule, ligand, or the first messenger reaches its target cell and binds to a specific receptor. This receptor then activates signaling cascade involving the intracellular enzymes and molecule, which is a second messenger, which again leads to a specific reaction. How? Via signal amplification. The number of signaling molecule is increased at every step of the signaling cascade. So, depending on the type of signal ligand, we have different type of receptors. What is a receptor? Receptor is a protein that receives signal from outside cell by binding specific ligand, initiating an intracellular signaling cascade. They are of two types. It could be cell surface receptor, also known as membrane receptor, or intracellular receptors, also known as nuclear receptors or IP3 receptors. So what is a ligand? Ligand is the first messenger that binds specifically to one receptor, and the type of ligand determines which receptor it will bind. So suppose we have a lipophilic ligand. Lipophilic ligand can pass through cell membrane, which is lipid bilayer. Hence, it can bind to the intracellular receptor. However, hydrophilic ligand cannot pass through lipid bilayer that easily, hence it acts on integral membrane receptors. Let's talk about intracellular receptors. They are of two types. IP3 receptor, which is present on endoplasmic reticulum, and the nuclear receptor, which is a ligand-dependent transcription factor. What happens is that a lipophilic hormone diffuses through the cell membrane. Then the hormone and receptor forms a complex. This complex then binds to the hormone-sensitive elements in the nucleus and acts as a transcription factors. Depending on the ligand, we have receptor present in cytosol or the nucleus. Let's suppose we have a mineralocorticoid as a ligand or glucocorticoid. Its receptor is present in cytosol. After binding the ligand, the receptor migrate as a complex with ligand into the nucleus. However, let's suppose we have thyroid hormone or vitamin D receptors. Their receptors are present in nucleus. They are specifically located in the nucleus that are already bound to DNA and are reached by their ligand directly. Then we have cell surface receptors. Cell surface receptors are of three types: the G protein coupled receptor, the enzyme linked receptor, and the ligand gated ion channels. G protein coupled receptors are the largest family of membrane receptors, and its action depends on three elements: the receptor, the G protein, and the effector molecule. For today's lecture, the G protein coupled receptors are more important, so I will focus on that. The receptor is a seven membrane transmembrane helicases, and it has a binding site for ligand. What happens is that the receptor has different kinds of subunit. It could be a GQ protein with alpha subunit. The protein could be GS protein, or it could be GI protein. And depending on the type of G protein the ligand stimulates, the effector molecule differ. Suppose we have a ligand which stimulates the GQ protein. The effector molecule for that is IP3-DAG. IP3 also known as inositol triphosphate, DAG is diacylglycerol. Suppose we have a ligand which stimulates the GS protein alpha subunit. The effector molecule is cyclic AMP. It will stimulate the production of cyclic AMP. Or suppose we have a ligand which stimulates GI alpha subunit. The effector molecule is again cyclic AMP. However, it will inhibit the cyclic AMP. Look at this diagram. This is the G protein coupled receptor. This is the G protein coupled receptor, and we have a ligand. For all kind of G protein coupled receptor, there is a general phenomenon. What happens is that suppose we have ligand, the ligand will bind to the receptor. This is the receptor. Now when a ligand binds to the receptor undergoes conformational change and it binds to the intracellular G protein. This is the intracellular G protein. As you can see, G protein is a heterotrimetric protein having three subunits: alpha subunit, beta subunit, and gamma subunit, right? Alpha, beta, and gamma. This is a heterotrimetric protein. So ligand binds to the receptor. This receptor undergoes a conformational change. When it undergoes a conformational change, it binds to the G protein, activating the G protein. The activated G protein will convert GDP into GTP. Okay. This activated G protein then dissociates into a complex of beta and gamma subunit and alpha subunit. Now, what you guys should know is that the G protein alpha subunit has an inherent GTPase activity, which hydrolyzes GTP into GDP and hence inactivated this G protein. Now, let's take a step back. This activated G protein can activate an enzyme known as phospholipase C. Now, phospholipase C, this is the phospholipase C enzyme. Its function is to cleave off membrane-bound lipids. One of the membrane-bound lipid is PIP2. PIP2, also known as phosphatidylinositol 4,5-bisphosphate. This will yield two products, IP3 and diacylglycerol. IP3 also known as inositol triphosphate, and DAG is the diacylglycerol. Now, recall that when we were discussing intracellular receptors, we also talked about IP3 receptors on endoplasmic reticulum. So what this IP3 will do, it will bind to those receptors present on endoplasmic reticulum and release calcium from endoplasmic reticulum via calcium channels, IP3 complex calcium channels. This will increase the intracellular calcium, causing the smooth muscle contraction. Another product that was yielded by this membrane-bound lipid cleavage was diacylglycerol. The diacylglycerol will activate protein kinase C, which is also calcium dependent, but calcium is coming from endoplasmic reticulum. So this protein kinase C along with calcium will be activated by diacylglycerol, and which will ultimately cause cell growth and proliferation. Now let's talk about different G protein. Now the G protein here is the GS protein instead of GQ protein. Now we have again a ligand binding to the coupled receptor. However, it is activating the GS alpha subunit. What it will do is it will activate the adenyl cyclase, which is an enzyme, which will convert ATP into cyclic AMP. Cyclic AMP will then bind to protein kinase A. Together, they will phosphorylate protein and generate a cellular response. Suppose we have a ligand. Instead of activating GS alpha subunit, it is stimulating the GI alpha subunit. Now, same thing, except the inhibition of this whole process will occur. Instead of stimulation, the inhibitory response will occur. Look at this diagram. We have all three G protein receptors. Focus on GQ first. So we have a ligand, which is a neurotransmitter acting on the G protein coupled receptor, activating a GQ protein. Now, GQ protein will cause the activation of phospholipase C, which will yield two products, diacylglycerol and IP3. IP3 will cause calcium release, and diacylglycerol will activate protein kinase C. Together, they will increase protein phosphorylation and activate calcium binding proteins. Then we have another ligand which is stimulating the GS receptor alpha subunit. This will activate adenyl cyclase enzyme, which will cause increased cyclic AMP, which will activate protein kinase A and will generate a cellular response, which is increased protein phosphorylation. Same thing, GI, except you can see it is inhibiting adenyl cyclase, reducing the amount of cyclic AMP, decreasing the activity of protein kinase A, leading to decreased protein phosphorylation. Let's discuss fluid compartments. 60% of our total body mass is composed of water. Of that 60%, majority of it is found inside the cell, which is intracellular. So 2/3 is intracellular, while 1/3 of the total body water is extracellular fluid. Of that extracellular fluid, 3/4 of it is found in the interstitium, while 1/4 of it is found in the plasma. So if you look at a fluid compartment of a 70 kg adult man, 60% of his body weight is water. That accounts for 42 liters. Of that, 28 liters is inside the cell, while 14 liters is extracellular. Then 1/4 of it is intravascular, which is in plasma, 3 liters, and the rest is in the extravascular fluid, could be interstitial fluid or transcellular fluid. Look at this diagram. This is the distribution of ions and cations. Now, ions can be divided into cations and anions. Cations are positively charged ions, whereas anions are negatively charged ions. You can see the division of anions and cations in the extracellular compartment and the intracellular compartment. Focus on this side of the diagram. You see this is the intracellular compartment. The major intracellular cation is potassium and magnesium. whereas major intracellular anions include phosphate and organic anions along with proteins. The major extracellular cation include the sodium and chloride, whereas major extracellular anions are chloride and bicarb. Fluid compartments always maintain same concentration of positive charges as negative ones. This whole concept is known as the principle of macroscopic electroneutrality. For example, the potassium in the intracellular fluid is balanced out by the negatively charged proteins and organic phosphates. So how do fluid volume shifts between the different spaces in the body? Recall that normal serum osmolarity ranges from 275 to 299 milliosmol per kg. There is an equilibrium established between cells uh, inside the cell and the extracellular fluid, right? Fluid shifts only if there is a difference in osmolarity between the compartments. Water flows from a compartment of low osmolarity to the compartment of high osmolarity. Why is that? If there is a high osmolarity, what is osmolarity? It is the amount of solvent, a solute over solvent, right? So if the osmolarity is high, the water will move towards it, which can typically manifest as edema, dehydration, changes in blood pressure, seizures, changes in intracranial pressure. Now, suppose you have a cell, and inside the cell you have an osmolarity of 299, right? There is an equilibrium among compartments. So if there is the osmolarity of 299 inside the cell, there is osmolarity of 299 outside the cell. There will be no fluid shift among compartment because there is an equilibrium. The example could be, let's suppose there is a patient after suffering from trauma, presents to you with blood loss. The patient is losing blood. However, there is no difference in osmolarity, right? Since there is no difference in osmolarity, there will be no shift of fluid. You can resuscitate patient using normal saline, which has same concentration as plasma, hence adding the isotonic fluid. Suppose you have another cell, but now its osmolarity is around 299, while the osmolarity outside the cell is around 200. As you can see, there is a difference. The osmolarity inside the cell is increased. That means the fluid inside the cell is more concentrated than outside the cell. Hence, water will move inside the cell. Right? Another example. Now we have the cell which has an osmolarity of 200, and the extracellular fluid has the osmolarity of 299. Now, the concentration outside the cell has increased, right? Now, water will move outside the cell. Let's discuss another concept known as effective circulating volume. What is effective circulating volume? It is a portion of extracellular fluid in the arterial system that effectively perfuses tissue. Its key regulators include sodium. However, this whole volume is modified by cardiac output, intravascular volume, and vascular resistance. You can estimate this effective circulating volume using blood pressure clinically. And the major regulators of this volume include sympathetic nervous system and renin-angiotensin-aldosterone system. Now, if you recall one thing, recall that your blood pressure is equal to cardiac output times total peripheral resistance, right? And cardiac output is stroke volume times heart rate. Using all these variables, you can modify the blood pressure, ultimately modifying the effective circulating volume. You should know that effective circulating volume not necessarily correlates with total body water. How? Let's suppose we have this uh, variable being compromised, which is cardiac output, yes, and it is compromised due to decreased intravascular volume. Recall that cardiac output is dependent on stroke volume as well. Suppose we have a patient with hemorrhage and he or she presents with volume depletion. So intravascular volume is down. That means cardiac output is down. That means blood pressure is down, right? Since cardiac output is down, stroke volume is down, total body water is also down, right? Total body water is down. But since cardiac output is down, leading to blood pressure compromise, the effective circulating volume is also down. Let's take another example. This time, we have a pump failure in such as heart failure. In heart failure, we have reduced ejection fraction, right? Which will ultimately lead to decreased cardiac output. Now, decreased cardiac output is due to heart failure, not because of decreased intravascular volume. The intravascular volume is normal. So, since cardiac output is down, the effective circulating volume is also down. However, since effective circulating volume is down, it will activate the major regulator, sympathetic nervous system, and renin-angiotensin-aldosterone system. These two will eventually cause sodium and water retention, causing the total body water levels to increase. So in heart failure, we will see increased total body water. However, the initial problem is not yet solved. Hence, the effective circulating volume will remain low. So in heart failure, you will see increased total body water while decreased effective circulating volume. Let's take another example. This time we are taking an example of cirrhosis. In cirrhosis, what we see is that there are two things happening simultaneously. First of all, the synthetic function is down. Synthetic function of liver is down, and one of the synthetic function is to synthesis of albumin. Albumin function is to maintaining oncotic pressure, right? So since there is decrease oncotic pressure, all the volume will be leaked into the interstitial spaces. That will activate the effective circulating volume. Not only that will decrease the effective circulating volume. Not only that, in cirrhosis, we have liver, which is releasing the major production of many vasodilators, especially nitric oxide. Now, these vasodilators will decrease the vascular resistance because it will cause vasodilation, including of splanchnic vasodilation. This decreased vascular resistance will lead to drop in blood pressure. When there is drop in blood pressure, it will cause decrease effective circulating volume. When effective circulating volume will go down, it will stimulate the system, which will cause retention of water and sodium. When water and sodium is retained, the total body water will increase. However, the vasodilators are still there, the vascular resistance is still down, hence the effective circulating volume will remain low. So in these two pathologies, you will see increased total body water and effective circulating volume. However, in hemorrhage, you will see decreased total body water along with effective circulating volume. Now let's talk about water homeostasis. Water homeostasis is the balance of osmolality in the intra and extracellular compartments, and it is maintained by osmosis and diffusion. Whenever we talk about water balance, we will also discuss sodium balance because water follows sodium. So water loss can be renal or extrarenal through skin, lungs, or GI tract. Water absorption occurs via GI tract. The key regulators of water balance include sympathetic nervous system. How does sympathetic nervous system is stimulated? We have baroreceptors. Suppose there is decrease or drop in blood pressure, baroreceptors will detect that drop and that will cause decrease firing frequency of those baroreceptors. This will cause decreased signaling to the brainstem, more specifically to the vasomotor center. That will decrease the parasympathetic stimulation and increase the sympathetic innervation. Increased sympathetic innervation will cause vasoconstriction, and the sympathetic innervation directly to the heart will increase the heart rate, contractility, leading to increased stroke volume, and ultimately, all these variables will increase the blood pressure. Other key regulators include antidiuretic hormone, which is stimulated when there is low osmolality and very, very low effective circulating volume. Then we have again sympathetic nervous system, which can detect drop in blood pressure. And we have renin, angiotensin, aldosterone system. So drop in blood pressure is essentially reflects the decreased effective circulating volume. And effective circulating volume is the volume that is effectively perfusing the tissue. One of the tissue is the renal tissue. So drop in blood pressure reduce renal perfusion. And if the pressure in the renal artery falls by more than 10 to 15 mmHg, proteolytic renin is released from juxtaglomerular apparatus. Now, renin converts angiotensinogen 1, angiotensinogen to angiotensin 1, and then angiotensin 1 will be converted to angiotensin 2 by the ACE enzyme. This angiotensin 2 can increase blood pressure in two ways. It can directly cause vasoconstriction because it is a very powerful vasoconstrictor, or it can cause the release of aldosterone, which will cause retention of sodium and water. So what is renin-angiotensin-aldosterone system? It is a hormonal system that regulates arterial blood pressure and sodium concentration. What happens is that we have afferent arteriole, which has baroreceptors. So if there is decrease or hypoperfusion of the renal artery, that can be detected by the baroreceptors. Other than that, the hyponatremia, which is the low sodium states, can be registered by the macula densa when sodium concentration in the DCT decreases. And then we have increased sympathetic tone. Suppose there is a drop in blood pressure and then there is a increased sympathetic tone, which can activate renal beta 1 receptors, causing the release of renin. So all these changes will cause release of renin by juxtaglomerular cells, which can lead to the conversion of angiotensinogen produced by the liver to angiotensin 1. And then angiotensin 1 is converted to angiotensin 2 via the ACE enzyme, also known as angiotensin converting enzymes, which is mostly produced by the liver. So look at this diagram. When there's drop in blood pressure, it will cause release of renin. Renin will convert angiotensinogen, which is produced by the liver, to angiotensin 1. Angiotensin 1 is converted to angiotensin 2 by the enzyme ACE, which is produced by the lungs. Now, angiotensin 2 has many functions. It can act on hypothalamus and cause you to feel thirst. It can directly act on the vascular smooth muscles and cause vasoconstriction. It can act on adrenal cortex to relieve and cause the stimulation of aldosterone. And then it can also cause the release of antidiuretic hormone. All of this can increase the blood pressure and vascular tone because volume will increase and vasoconstriction will also cause the vascular tone to increase. Let's talk about angiotensin 2. It acts as a strong vasoconstrictor. It desensitizes the baroreceptor to hypertension, preventing reflux bradycardia. It stimulates thirst in the hypothalamus. It increases ADH secretion from posterior pituitary. It induces the secretion of aldosterone by the adrenal cortex. And aldosterone increases the renal absorption of sodium and water, which will lead to increased extracellular fluid, increased blood pressure, decreased potassium, decreased hydrogen ion, and ultimately causing alkalosis or increased pH. So initiation of thirst can be stimulated by two mechanisms. So neurons in hypothalamic regions usually stimulate thirst. Stimulation can be directly due to increase in osmolality. The osmoreceptors will detect it in the hypothalamic region and will cause the initiation of thirst. Or we have angiotensin 2, which can directly stimulate the hypothalamic neurons, causing the thirst. What are the overall effect of this system? It will cause increased arterial blood pressure and increased blood volume. In renal, it will maintain renal function and volume stasis in the low volume states. Increased vasoconstriction of the afferent arteriole. Efferent arteriole causes effective filtration pressure and filtration fraction, which will help maintain GFR during renal hypoperfusion. If you look at it, this is the afferent arteriole and this is the efferent arteriole. Vasoconstriction over here will cause the blood to forcibly move into the glomerulus, right? So it will maintain what? GFR and increase the filtration fraction. Not only that, maintenance of GFR during hypoperfusion, which is achieved by increased vasoconstriction of the efferent arteriole, cause the increased effective filtration pressure and increased filtration fraction. There will all be a compensatory increase in sodium absorption as well in the proximal convoluted tubules and distal convoluted tubules, preventing net volume loss. Another one of the hormone released is the antidiuretic hormone, which is regulated by the plasma osmolality. This is the major trigger of it. The osmoreceptors in the hypothalamic region will detect it and then will cause the release of ADH. Hypovolemia via the sense by the atrial stress receptors will decrease the ANP production, which counteracts the ADH. So that is how ADH is regulated by the hypovolemia. Hypotension sensed by the peripheral baroreceptors, and angiotensin 2 can directly stimulate the ADH release. What are the effects? So, antidiuretic hormone can regulate plasma osmolarity, which is mediated by V2 receptors. Now, V2 receptors will cause insertion of aquaporin channels in the principal cells of the renal collecting duct and distal convoluted tubule. This results in increased water absorption, which is lead to free water absorption. What I want you guys to know is that V2 receptors act via GS protein mechanism. So you have an ADH, which will, you have what? You have ADH. ADH will act on V2 receptor, and this V2 receptor essentially works by stimulating the G protein couple receptor, but more specifically GS alpha subunit. Now, GS alpha subunit will stimulate what? Adenyl cyclase, which will convert ATP to cyclic AMP. And then cyclic AMP will activate protein kinase A, right? And then generates its response. This is very important concept. Another way ADH can regulate blood pressure is can exert its effect is through regulation of blood pressure. However, this is mediated by V1 receptors. Now, for V1 receptors, for V1 receptors, again we have G1 protein, GQ protein couple receptor is going to be stimulated. Like for example, we have ADH. ADH will stimulate V1 receptor, and this will work via stimulating the G protein, but this time we have GQ alpha subunit being stimulated. This will cause the phospholipase C enzyme to get activated, which will release what? Inositol triphosphate, IP3, and diacylglycerol, right? IP3 will cause calcium release from endoplasmic reticulum, which will cause smooth muscle contraction, right? So you see how it is causing vasoconstriction. Okay. Next, vasoconstrictive effects are at higher temperature. It can also increase urea absorption in the collecting ducts, increases the corticomedullary gradient, and facilitates the urine concentration. It can also cause ACTH release. So this is the negative feedback loop. As you can see, water homeostasis. What happens is that suppose there's dehydration. In dehydration, what happens is we have decreased water and increased sodium. That means there is an increased blood osmolality. Blood osmolarity will be detected by the osmoreceptors in the hypothalamus. This will cause the release of ADH from posterior pituitary gland. Not only that, it can directly stimulate thirst, and you will drink water, and ultimately water levels will increase and negative feedback will stop this whole process. However, ADH will cause stimulation of distal convoluted tubule and collecting duct. It will insert what? The aquaporin channels and increase the water reabsorption. This will cause reduced urine volume and increase sodium water, the ratio of sodium, water, and urine, which will negatively uh, that will decrease the sodium concentration and will stop this whole process. Let's talk about aldosterone now. How does aldosterone work? Now, aldosterone is a steroid hormone, very important to understand this, also known as mineralocorticoid. It is released from adrenal gland. It binds to the intracellular mineralocorticoid receptor. Like I said, it is a steroid hormone. It can cross partially permeable membrane easily. Hence, its receptors are located inside the cell. Now, if you recall correctly, intracellular receptors could be in cytosol or it can be inside the nucleus, but for mineralocorticoid, the receptors are located in the cytosol. The complex will form and it will go inside the nucleus and exerts its transcription factor wala effect. Now, look at this diagram. If you see, this is the aldosterone entering the cell because it can cross partially permeable membrane. It has what? Cytosolic receptors present. Then it will, this whole thing, this complex will move inside the nucleus and cause transcription. Transcription and then translation. Translation of different receptors will occur. First, different pumps and receptors. First, there will be increased pumps of sodium and potassium. This will cause sodium release into the interstitium and potassium reabsorption. When this happens, the concentration of sodium inside the cell decreases, right? Not only that, it is increasing the sodium channels in the apical membrane as well. So inside the cell, the sodium concentration decreases. So what sodium will passively move into the cell through ENaC channels. We have also increased translation of ROMK channels, which can allow the potassium, which is entering inside the cell, to leave through tubular lumen. And as you all know, water follows sodium. So wherever sodium is going, water will follow it. Not only that, translation of hydrogen ATPase pump will also occur. This will allow the hydrogen excretion. So the net effect is there will be decreased sodium in urine, increased potassium excretion, decreased water excretion, and increased hydrogen ions, which will lead to decreased pH. Inside the interstitium, what will happen? We will have sodium and water reabsorption. We will have hypokalemia and we will have increased pH. This is the net effect of aldosterone. So let's read out what aldosterone is doing. Firstly, it is increasing sodium potassium ATPase pump in the basolateral membrane, which will increase transportation of sodium out and potassium into the tubule cells. It is also increasing apical hydrogen pumps, which is excreting the hydrogen ions. Then it is also increasing sodium channels in the apical membrane area, which is causing increased sodium reabsorption. And we have ROMK channels, also known as renal outer medullary potassium channels, which is facilitating the potassium excretion. So what are the net effects? Sodium absorption along with water reabsorption and potassium excretion into the urine along with hydrogen excretion. This will increase blood pressure, which will cause hypokalemia, and it will increase the pH levels. Clinical significance is dehydration. Causes include sweating, decrease water intake, diuresis or osmotic diarrhea. Fluid loss could be hypotonic, that such as include free water loss. It occurs from all water bodily fluid compartments. Intracellular compartment is most affected, which leads to decreased effective circulating hypertonicity. How can it be compensated? By activation of thirst centers, ADH release. In hypovolemia, dehydration versus hypovolemia, the effective circulating volume is depleted, right? The causes include vomiting, inflammatory diarrhea, and secretory diarrhea. Fluid loss is typically isotonic. In dehydration, the fluid loss is hypotonic, but in hypovolemia, the fluid loss is isotonic. So it will primarily, it occurs primarily from extracellular compartment, right? So intracellular compartment is mostly unaffected, typically does not affect extracellular fluid tonicity. It can progress to shock if severe. The compensatory mechanisms include RAS activation and ADH release. Thank you.